Hi, welcome back to Bio23 Infectious Disease for another chapter where we're going to be talking about the concepts of microbial disease, learning a little bit more about the basic foundations of what allows microbes to cause infections. We're going to start off on chapter 10, learning a little bit about microbial associations. Microbes can associate with humans in lots of different ways. One of these ways is a symbiotic association. Many people misdefine symbiotic association and define it as an association between two organisms where one organism is benefiting another organism and they're mutually beneficial. That is not, in fact, the definition of symbiosis. A symbiosis is when two organisms are interacting and at least one of those organisms requires that interaction for survival. In the case of microbial biology, it's usually the microbe that requires that interaction for survival, and the host, in this case us, that does not. Within this general category of symbiosis, there falls three different categories. There's mutualism, commensalism, and parasitism. Mutualism is when two organisms are interacting in a symbiotic association, so that interaction is required for at least one of the partners' survival. But the mutualism relationship is when both partners are benefiting from that association. The classic example of this is an interaction between the microbes in the intestine and the living organism. For instance, termites, which eat their way through wood, are not able to digest the cellulose or the wood itself were it not for the microbes that live in their gut. It's the microbes, in fact, that are responsible for breaking down the cellulose in the wood. The termites cannot do this alone. Similarly, the microbes in our intestines help us break down our food as well. Famously, for a mutualistic relationship is the interaction we have with the E. coli in our intestines. I'm aware that many of you associate E. coli with negative interactions and food poisoning, but in fact, everyone has E. coli in their intestines, and the E. coli in our intestines help us digest our food, obtaining nutrients from the food, as well as obtaining nutrients that are generated from the microbes themselves. In particular, E. coli makes vitamin K for us. As you may consider, vitamin K is not one of those vitamins that you hear about too much. That's because we're not in desperate need of vitamin K, because the E. coli always provides it for us in plenty supply. The second interaction that we have in a symbiotic association could be a commensalism. Commensalism is when one organism benefits and the other is unaffected by the interaction. There are many commensal relationships that we have with lots of different microbes in our body. For instance, this chart that you see here shows you all the different types of microbes that we have in and on our body, and also the association. Most of these microbes are commensals and live in and on our body, eating off of our dead cells or food that we are unable to eat, thus not harming us by their growth, but also not helping us either. Finally, there is a third type of interaction, and that's parasitism. This type of interaction is when one of the partners in the symbiotic association benefits, and the other one is harmed by that interaction. I'm sure you've heard of parasites before, and probably know that it's usually the microbe that's gaining the advantage and the host that is suffering the cost. There are lots of really common examples of parasites. Depending on how you define them, some people define parasites as just including protozoa like Giardia or malaria, and helminths like tapeworms and pinworms and things like that. However, there are some biologists that also consider fungi and bacteria can be parasites as well. This would include athlete's foot, roundworm, which is actually a fungus, and also salmonella, cholera, shigella, and other types of pathogenic bacteria. Now, while some microorganisms might be classified as being mutualistic or commensals with the human body, that association can change if the human body changes. These categories are not set in stone. As your book describes, if the immune system for a particular human were to become depleted or depressed, that could allow for non-pathogenic microbes normally engaged in a mutual or commensal relationship with us to transition into a parasitic one. We see this, in fact, quite often with HIV and AIDS patients. When their immune system is depressed and not able to fight off infections, previously non-pathogenic microbes growing on the skin can begin to invade the skin, generating bed sores and open sores. Other alternatives are when burn victims can be at major risk of serious bacterial infection because their skin no longer provides a sufficient barrier for pathogenic invasion. These examples are all examples of what are referred to as opportunistic pathogens or facultative parasites. Opportunistic pathogens or facultative parasites are ones that are not normally pathogenic, but when the immune system becomes depressed and the situation allows for it, the pathogens can become pathogenic and invade the body. We can also see that microbes that are non-pathogenic can become pathogenic if allowed in new areas of the body where they used to not previously be. This happens where if in a colonoscopy or surgery, there is a tear in the intestinal lining of the large intestine, allowing microbes to escape the large intestine and get into the bloodstream and the body cavity. This is an example of peritonitis and can be quite deadly fairly quickly within just a few days. We see that here. These microbes, like the E. coli that normally live in our body, are non-pathogenic. But if the appendix ruptures or there's a tear for another reason, that is when death can easily occur. Now, while most of our discussions this semester will be focused primarily on bacteria living by themselves, in fact, bacteria talk to each other quite regularly. One of the ways they can do this is using the process known as quorum sensing, which we talked about in a previous chapter. Quorum sensing is when one microbe talks to other microbes of the same species. Remember, bacteria are all clones of each other, and because they're all clones of each other, they're essentially talking to themselves. When they register that there are enough of them around, as indicated by the concentration of inducer molecules that they are constantly secreting or releasing from their cells, they then shift into a different behavior. This different behavior could be innocuous, including things like bioluminescence or just glowing, which we see in some jellyfish bacteria in the oceans, or fermentation, like when we have fermentation of foods. Or it could be more deadly in the case of some pathogens that only decide to attack the host when there is enough concentration of them around in order to attack sufficiently, defending themselves from white blood cells in the body. That is a type of interaction or communication known as intraspecies communication, where cells of the same species are talking to each other. That's different from interspecies communication, which is when two different cells of different species talk to each other. In the intraspecies communication known as quorum sensing, the microbes won't decide to attack the human body until there are enough of them around. Once they determine that there's enough of them around, they'll then shift to a pathogenic state, and in doing so generate what's known as virulence factors. Virulence factors are genes or proteins that are used to cause disease. Remember that the definition of virulence is the degree of pathogenicity, or how pathogenic or disease-causing a microbe is. Alternatively, microbes trying to cause an infection can shift from a more mobile state to the more sedentary state known as a biofilm. Quorum sensing can also lead to biofilm formation in that when the microbes notice there are enough of them around, they can all shift to form that sedentary, very sticky, very thick growth of bacteria and other microbes. Creating a biofilm requires a community of microbes, and thus is heavily regulated by quorum sensing. Those cells in a biofilm exude extracellular polysaccharide polymers, or EPS, which are really sticky sugars that coat the entire growth and can protect the bacteria from things like disinfectants and antibiotics, as well as keeping them intact and protected from your immune system. As is obvious, quorum sensing and biofilm formation can be quite an advantage to infectious bacteria. That's why there's research aimed at minimizing or eliminating the ability of microbes to use quorum sensing. In particular, antibiotic-resistant strains of bacteria, which resist antibiotics, are very difficult to treat. However, the discovery that a number of these antibiotic-resistant strains also use quorum sensing have allowed scientists to target the quorum sensing aspect of the infection rather than the bacteria themselves. There's been a lot of research in learning how to silence this communication between the microbes, keeping them from being able to interact and communicate with each other. By shutting off quorum sensing, this allows for bacteria to no longer communicate their numbers and to no longer be able to detect how many of them are in the area. And as a result, this keeps the microbes from generating disease, even if they are antibiotic resistant and growing in the body. While this technique has been demonstrated in research labs, unfortunately, there are no quorum sensing inhibitors that have been approved by the FDA for use in the United States to treat antibiotic resistant strains today.